So thanks everybody for coming today on this online version of the Conservation Sciences Seminar Series. I'd like to thank Ed Swain for being flexible in presenting about wild rice and sulfate. So Ed is an adjunct assistant professor in the Department of Fisheries, Wildlife, and Conservation Biology at the U of M. His research is focused on the effect of sulfate pollution on aquatic systems, especially how sulfate affects wild rice and mercury methylation. In 2019, he recently retired from the Minnesota Pollution Control Agency after 31 years with the agency. And he's continuing his career by helping students at the University of Minnesota. So thanks, Ed. And just an announcement to people, if you have questions about the talk you'd like Ed to answer, you can go ahead and type those into the chat box during at any point or afterwards. And then at the end, I'll read them aloud and then Ed can answer all the questions. So go ahead, Ed, thank you.
It might be a good idea for everybody to mute and stop their video to give Ed a little more bandwidth. In previous Zoom meetings I've been on, that seems to help.
Okay, thanks, John. Thanks, David. So I'm going to talk about the old sulfate standard to protect wild rice and the Pollution Control Agency's efforts to update it that I was involved in and what happened to that. Let's see. There. So this story about the sulfate standard to protect wild rice is a long story with a lot of history and a lot of baggage. And so I'm going to talk a lot about all this history, and I've got 72 slides. So I'm going to move right along. And I've developed a structure that we'll see these slides for each of these 12 sections as we go. Note that in the bottom right-hand corner, there's a number on the slide. If you have a question about a particular slide, note that number and we can jump back to it when there's time for the questions. So the ecology of wild rice, there's one species in Minnesota, Zizania palustris. It's an annual plant. It germinates after spring thaw and matures in late August. The plants mature the seeds, drop them into the water, they sink into the mud, and then they stay there over the winter, and they have to be cold all winter, or else they won't sprout in the spring, close to freezing. And then they sprout and do it all over again. And it can form extensive monocultures in Minnesota. Minnesota is really the center of this species of wild rice. The floating leaf stage in June, when the leaves come up and are floating on the surface, is vulnerable to a sudden increase in water level, which pulls the small roots out of the sediment. And there's a 4 to 6 year boom-bust cycle with a slow release of nutrients, especially nitrogen, that John Pastor has demonstrated from the decaying roots and straw. Wild rice can occur with perennials like water lilies, but under stable water levels, the perennials may win the competition for space. So ecologically, wild rice is an opportunistic species. It's an annual, it perhaps needs disturbance to fend off the perennials, which could dominate the space. And typical disturbance might be the sudden levels in lake levels or drying out, something that beats back the perennials so that the wild rice can come back in. In 2008, at the instruction of the legislature, the DNR issued a big report on wild rice, and I think this map came out of their effort to document all the water bodies in Minnesota that have wild rice. Wild rice is important in Minnesota. It's an important ecological and economic resource. It's got importance culturally and spiritually, especially for Native Americans. And the legislature, in their wisdom, officially made wild rice Minnesota's state grain. Zizania palustris, the species that needs to be cold in the winter, historically had a big range into Canada, Minnesota, east to New England, where it stopped. There's a similar species, it looks similar, but it's Zizania aquatica, which is ranges from Wisconsin east to southern New England, then all the way down the Atlantic coast to Florida and into Louisiana. And it looks very similar, but it does not require the cold winter. And then there's a rare species, Zizania texana, that's only in one stream in Texas. So there's 3 species of Zizania in North America. There's one species in Asia that we won't talk about. So here again is the life cycle of wild rice, starting with that seed down in the mud that fell there the previous fall. In the spring, it germinates, roots go down, the stems go up, get a floating leaf stage, and eventually get aerial stage and male flowers and female flowers above that. They don't normally self-pollinate. They need to cross-pollinate. They make the seeds fall into the water and the mud, and that starts all over again. Here's the floating leaf stage, and it's gathering photosynthetic product, sending it down to the roots so it can grow better roots, and then it grows the aerial stage after that. And it can have multiple stems, or called tillers, and it drops all these seeds, which can be harvested by ducks and humans. And here's 2 men from the Leech Lake tribe harvesting wild rice in the only way that's legal in Minnesota on public waters. There are also cultivated paddies that are developed on soil, upland soil, but that's private property. And public waters, this is the only legal way. It's when the grains are ripe, the one person in a canoe leans the plants over the canoe and brushes it with a knocker, and somebody else in the canoe pushes it along, and the rice falls into the bottom of the canoe. Okay, now second part of the talk is about John Moyle. John Moyle is important because he's the origin of the sulfate standard that was put in place. His 1939 PhD thesis was titled "The Larger Aquatic Plants of Minnesota and the Factors Determining Their Distribution." And what when I worked at the Pollution Control Agency, would hear sometimes is, oh, this is old science. It wasn't very good because it's old. And I want to counter that he and his fellow graduate school mates were very good scientists and very sophisticated. One of his fellow students was Ray Lindeman, and they were friends. They did field work together. They acknowledged each other in their publications for helping with their field work. Ray Lindeman wrote one of the most important papers in ecology, where he proposed that it was important to track energy flow through ecosystems. The paper was published after he died at the age of 27, and I checked yesterday at Google Scholar, it's been cited over 4,000 times. I think Lindeman and Moyle influenced each other quite a bit. And Moyle went on to have a long, productive career at the Minnesota DNR, where he showed that he had a very good ecosystem sense. They're both these people and their other students were great naturalists, they were great observers, and great scientists. And we just need to remember that. Here's a picture of when Moyle was older. In the 40s and 50s, he published several papers about wild rice. And in a 1956 paper, he mapped his conclusion that wild rice occurrence is correlated with low sulfate waters. Wild rice limit that he drew, that green line, what he was saying is that wild rice was mainly to the northeast of that, where the waters, a lot of the waters were below 2 milligrams per liter sulfate, and virtually all of them below 10 milligrams per liter. So it was an observation on his part. At that time, he didn't really know what might be cause and effect, but he published it. And as a result of his observation, it was confirmed later during the Pollution Control Agency research, where we mapped the DNR's wild rice occurrences over modern sulfate data. And you can see that in northeastern Minnesota, indeed, is very low sulfate. That blue is less than 5 milligrams per liter. Now there's an island in northeastern Minnesota of higher sulfate, and those are the waters that are somehow affected by the iron mining in northeastern Minnesota, where the waste rock had a little bit of sulfide minerals in it that oxidized the sulfate and raised the sulfate concentrations. And so based on his observation, in 1973, the Pollution Control Agency established a standard to protect wild rice of 10 milligrams per liter. The phrasing of the standard is 10 milligrams per liter applicable to water used for production of wild rice during periods when the rice may be susceptible to damage by high sulfate levels. Well, there's a couple of problems right away with that standard, aside from not knowing why the sulfate might be harming the wild rice. They said it only applies to wild rice waters, which they don't define. Well, what's a wild rice water? How do you know when you're in a wild rice water? And they also don't define what's the time period when sulfate could damage wild rice. So it wasn't a very good standard. It wasn't written very well. But this is in the early days of the Pollution Control Agency. 1973, the agency had been established in 1968. The US EPA wasn't established until 1970. And then the Clean Water Act, the Federal Clean Water Act, under which all these water quality standards were valid, had just been passed by Congress. So it's kind of understandable that the standard was written pretty loosely. And in 1940, John Moyle had published a paper where he wrote, no large stands of rice occur in water having sulfate content greater than 10 parts per million, which is the same thing as milligrams per liter. And rice generally is absent from water with more than 50 parts per million. So he published this correlation several times. Now I want to tell you all about sulfate, because you need to know about that in order to understand what happened later. Sulfate's a negatively charged ion. It's very common. In Minnesota, it ranges from less than 1 milligram per liter to over 700. It's not very toxic at all. It's pretty stable. It's common like chloride is common, and it more affects things by how it affects overall salinity rather than being a toxic agent on its own right. It's got one yellow sulfur atom surrounded by 4 red oxygen atoms, and it's got a negative charge of -2. And the 10 milligram per liter sulfate standard for wild rice is, say, in contrast to the water quality standard for drinking water of 250, which really isn't even based on toxicity. It's based on the people who aren't used to concentrations that high have trouble with the digestive system, but they can get used to it. It's really advice. It's not a hard standard. An important thing about sulfate, if you want to get it out of water, it's really difficult because it's non-reactive. You can't filter it out. About the only practical way to get it out is through reverse osmosis. And it's currently really the only reliable technology available to get down to low levels, close to 10 milligrams per liter. But it's extremely expensive, not only because of this equipment with all the membranes, but because it produces a salty brine that has to be disposed of. And what do you do with that brine? If you can't get rid of the liquid brine, you have to turn it into a solid by heating it up and boiling away the water. And that is expensive for the equipment. It's expensive from an energy point of view. For instance, for a moderate flow of 0.36 million gallons per day on the right, it's cost $12 million to build the facility and $3 million annually to operate it. So it's expensive to try to remove sulfate from wastewater. And then you have to landfill that waste salt that precipitates. And if you're trying to add this to a wastewater treatment plant, it's a whole new operation that's completely different from anything else. So the Pollution Control Agency concluded that if you had to have treatment, it would be unaffordable, especially for cities, municipal wastewater treatment plants. From a pollution regulatory point of view, under the Federal Clean Water Act, it's unaffordable. And it would automatically result in a variance. A variance is granted to a discharger when the discharge is unaffordably to treat. It's not permanent. There's an assumption that sometime something cheaper will come along that would make it affordable. But it's kind of a pain in the neck. Anybody who wants to get a variance has to apply for it. And that means they might have to hire a consultant to build the case that it's unaffordable. And then the PCA has to approve it. And then the US EPA has to approve each variance. An important thing about sulfate is that it can be converted to sulfide. Sulfate's on the bottom here in this chart. It's fully oxidized. It's got 4 oxygens on it. You can't get any more oxygen on that sulfur atom. The opposite of oxidized is reduced. And in chemical terms, it's not really the oxygen that makes the difference. It's how many electrons are on the molecule. As sulfate is reduced, electrons are added until you get to hydrogen sulfide, which has a lot more electrons, and it's highly reactive. And because it's highly reactive, it's toxic. And the main reason it's toxic is that it loves to react and bind with any metal. It likes iron, mercury, zinc, copper. And because it binds really strongly with these metals, some metal deposits are metal sulfide deposits. And these are the deposits in northeastern Minnesota that are currently being considered for mining, the copper nickel deposits. They're sulfide minerals. Not only can sulfate be converted to sulfide, but sulfide can be converted to sulfate if it's exposed to oxygen, and it goes backwards. In between is sulfur dioxide, which is often emitted from coal burning power plants as a gas, because there's a little bit of sulfur in coal, and when the coal's burned, it makes sulfur dioxide, which is quite toxic on its own right. But in the atmosphere, the traditional way of dealing with that pollutant like that is to build a tall smokestack and dilute it, and also to give it time to oxidize further all the way to sulfate in the atmosphere. And that makes it less toxic, but also happens to produce acid. So that's the origin of acid rain, when sulfur dioxide converts to sulfate, which is sulfuric acid. Anyway, these conversions of sulfur between sulfate and sulfide are important. So in the environment, the main way that sulfate is converted to sulfide is by anaerobic bacteria that can't get access to oxygen, so they essentially are breathing sulfate. And the sulfate can diffuse from the overlying water into the mud. And the reason the bacteria need the sulfate is they're extracting energy from the organic carbon. The energy is electrons. And the bacteria take in the carbon, strip off energetic electrons, then they have less energetic electrons they need to get rid of. And what do they do with those electrons? They give it to the sulfate, and that converts the sulfate to sulfide. And so that's how sulfide accumulates in the mud of a water that has sulfate in the overlying water. So now I'm going to tell you about all the enforcement of that 1973 standard that was established based on John Moyle's correlation. And all those enforcements constitute one time. The standard was put in a permit just once in 1975 for the Boswell coal burning power plant in Cohasset, Minnesota, just upstream from on the Mississippi River from Grand Rapids. in 1973, a bigger unit was added to the Boswell coal burning power plant, a huge unit, really, at 362 megawatts, which made it one of the biggest power plants in the state. And to combat that SO2, the pollutant that would go up the smokestack, the sulfur dioxide, they used a wet scrubber to remove it, and that went into an ash pond that discharged into the wild rice-rich Mississippi River. So this was well-meaning to capture the sulfur dioxide. That was important, but unfortunately it converted the air pollutant to a water pollutant, and it went into the Mississippi. So here's a current view of the Boswell plant on the Mississippi with the tall smokestacks. And here on this view, in the upper right are the ponds where the scrubber water goes with the ash and the sulfur dioxide, and which has been converted to sulfate. Here's the plant itself with a small, tall stack. Here's the Mississippi River is flowing by from top to bottom. And because they're using wet scrubbers, there starts to be too much water in the pond, so they discharge that water down this channel into this backwater of the Mississippi, and then it joins the Mississippi. And all this green around the Mississippi upstream, that's wild rice. So a lot of wild rice here. Well, Minnesota Power didn't want to remove and control the sulfate, probably because it would cost a lot. Plus, they just put on this wet scrubber to remove pollution. And so they appealed the inclusion of that sulfate standard, and there was a hearing, and the hearings officer heard contradictory expert testimony. Minnesota Power, which owned the Boswell coal burning power plant, hired Professor Stewart from the University of Manitoba, and he had observed wild rice growing fine at 200 milligrams per liter in Canada. And MPCA's expert, John Moyle, just repeated his Minnesota observations. But he also noted in his memo that there'd been a recent article in the journal Science that concerned the toxicity of sulfide in the mud to white rice. Now, white rice is pretty similar physiologically to wild rice. They aren't the same thing. They diverged over evolutionary time about 17 million years ago, according to one paper, but they operate about the same. So he wrote that it's probably the sulfide is the problem. So he understood that, but he was just retiring from the DNR about that time, and he wasn't involved after that. And the result of Minnesota Power's appeal was that the standard was changed from 10 milligrams per liter to 40 milligrams per liter downstream from the plant. And they decided that the time that it was important to have low sulfate was between April and June. I don't know how that was decided. And it was 60 milligrams per liter for the rest of the year. And as a requirement, Minnesota Power had to conduct a study of the effect of sulfate on wild rice at the site. And a big report was issued by Stewart and his graduate student, Peter Lee, in 1978. Peter Lee went on to publish about 30 papers on wild rice, and he's still a productive professor in Canada. And after that, the Pollution Control Agency didn't enforce the standard, didn't try to put it in any permits until about 2010. And I'm just guessing that there was an institutional memory at the PCA that, that there was a problem. And just as a note, downstream of the Boswell plant, currently still there's big beds of wild rice. The wild rice is doing fine downstream Boswell. And the PCA sent field crews there to look at it, and they even had trouble canoeing through the stands. They're so dense. And so as a result of requests to the PCA to put the sulfate standard in permits. Around 2010, the PCA started trying to do that, and industry started complaining. And in 2011, the Minnesota legislature directed the PCA to study and revise the sulfate standard. This was with industry support, because there was a feeling that 10 milligrams per liter was just ridiculously low, and any study that would collect data would show that that the sulfate standard could be a lot higher and protect wild rice. So the legislature gave the PCA $1.5 million and a pretty short time frame to spend it and collect the data to the end of 2013. And that's when I got involved, because I'd been working on sulfate, both in terms of acid rain and how it affected mercury methylation. I was sort of pre-adapted to work on wild rice. And the PCA anticipated that a study was needed, and even before the legislature awarded the money, two months before that, the PCA sponsored a conference with 36 experts from Minnesota and actually federal people from across the country to talk about how sulfate could affect wild rice. And this diagram was discussed at that meeting. The conversion of sulfate to sulfide is complicated and has a lot of secondary, tertiary consequences. And a lot of different things could be going on. Yet in the end, it came down to a simple hypothesis that sulfide was probably toxic to wild rice. And this is what needed to be assessed in the study. But there's lots of other sub-hypotheses, and so a lot of data was collected to try to assess those hypotheses too. So with that $1.5 million, PCA managed the study and contracted with the University of Minnesota, the Science Museum, and contracted with the Minnesota Department of Health Laboratory to perform a lot of analyses. Fortunately, John Pastor at the University of Minnesota Duluth had been interested in wild rice for a long time before PCA started researching it. And he had a lot of experience and a lot of knowledge about wild rice. And in fact, he had started working on wild rice, the sulfate issue, with the tribal support. So he was a natural person to work with, and he agreed to take on some of this research. In particular, he performed laboratory experiments, including hydroponic exposures of wild rice in those little bottles. And there's a new young assistant professor, Nate Johnson, in the engineering department. This didn't know John Pastor, but we put them together, and they've been working together ever since, very productively. And John Pastor had also started some outdoor mesocosm experiments with tribal funding, which the PCA augmented and kept going. And then since the PCA money ran out, John's and Nate Johnson have been keeping it going with other funding. So, and then most of the money actually went to a field survey of over 100 sites that was led by researcher Amy Murbo at the University of Minnesota Twin Cities campus, with support from Dan Engstrom from the St. Croix Laboratory of the Science Museum, and Jeff Brenner, who supervises the Minnesota Department of Health Laboratory. I think Jeff told me once that they, as part of this study, they analyzed over 60,000 samples. So there's a lot of samples, a lot of analyses. The field study analyzed lots of things in the surface water, and then the sediment analyzed the solid sediment called bulk sediment, but also extracted pore water from the sediment. This is the dissolve phase that maybe the roots would be interacting with more than the solid materials. So there's a lot of analyses of the pore water, including the sulfide in the pore water and lots of other dissolved things. And then other sediment properties and other variables were measured. And in some, every time a field crew went to a lake, they quantified 64 variables. Here's John Pastor leading a field trip of the advisory committee to the Pollution Control Agency's efforts. And he's showing them the outdoor mesocosms. You can see the floating leaf stage here, and then later in the summer, the wild rice emerged and grew the seeds. So they had 5 different sulfate concentrations in these mesocosms with 6 replicates at each concentration. It's quite a powerful experiment. And in the end, it was clear that when you add sulfate to a mesocosm, the sulfide here on the left increases a lot. And also at the same time, that sulfide's reacting with metals. And the main metal that's available is iron. And the iron would go down as the sulfide went up because they precipitate each other. And it was really clear that when you add high concentrations of sulfate up to 300, after a couple of years, the plants don't make seeds, and then because there's no seeds, the plants die out, plus it's not good for the plants. And an interesting observation that John Pastor and his group made is that at the high concentrations in the 300, the iron sulfide starts precipitating on the outside of the roots. And he had a hypothesis at the time that that iron sulfide was blocking availability of important nutrients, especially nitrogen, which is the limiting nutrient. And that was a hypothesis at the time. And he's been following up on that with especially a graduate student named Sophie LaFond Hudson and with Nate Johnson and others. They've published several papers since the PCA abandoned the whole rulemaking process. This upper paper was just published a couple of months ago, and then that's for Sophie's PhD. And the bottom paper was for a master's project. And then I just learned from John Pastor that another paper is coming out pretty soon. So they've had a very productive pursuit of this phenomenon of the black roots. And then the PCA took all this data from the 3 different efforts, the hydroponic, mesocosms, and the field data, and wrote a preliminary report, and it was paid for a peer review panel to look at it. 7 experts from around the world, really. Only 2 Minnesotans. Dr. Pat Brzonek was the chair, and then Dr. Sue Gladowicz, who's the chair of Department of Fisheries, Wildlife, and Conservation Biology at the university, was on it. She's an expert on wetlands. And then this whole issue of sulfate pollution affecting aquatic plants should have been pursued in Minnesota because of this issue with the wild rice, but it wasn't actively. However, it was pursued in other parts of the world. Dr. Gertie Arts from the Netherlands, because the Netherlands had documented toxicity of sulfate through sulfide on wetland plants in the Netherlands. And then there was 2 people from Florida on the panel, Dr. Don Axelrod and Dr. Curtis Pullman, because in the Everglades, there's an issue of sulfate pollution affecting the sea of grass, the sawgrass, and how the sulfate likely was turning into sulfide and poisoning the native sawgrass, and the cattails were moving in. As a result, and cattails are much more resistant to sulfide. And then Dr. Mark Hansen from Manitoba and Siobhan Fennessy from Kenyon College. So they were a really good panel, and they reviewed that preliminary analysis. And the preliminary analysis had asserted that dissolved sulfide is controlling the wild rice presence, and the analysis emphasized the control by iron availability. And the panel identified the field data as particularly powerful and recommended to reanalyze the data using different statistical tools, especially structural equation modeling. And they also recommended including organic carbon as a possible factor controlling sulfide. And then as a result of their advice and additional work, there was 4 papers were produced in 2017 based on this research. The first one was headed by John Pastor on the hydroponic and mesocosm data that he produced. And it showed that sulfide is toxic to wild rice. And the more sulfate you have, the more sulfide you get. A second article was headed by Amy Mirbeau on the field data that she managed. And that paper showed that the presence of wild rice is primarily controlled by dissolved sulfide. And the way that was demonstrated was using a tool called binary logistic regression. And this is a tool that has been used quite often in conservation biology when conservation biologists are interested in what habitat is suitable for a given species, especially endangered species. How do you identify habitat you need to protect a species? Because the main threat to species in the environment is habitat loss. So if you can identify what habitat the species needs, then you can protect the species. And so the way this works is that a statistical test called binary logistic regression is performed on a parameter like pore water sulfide, and it demonstrates the probability of the species, wild rice in this case, being present across the spectrum of concentrations in this case. So as pore water sulfide gets higher, the probability of wild rice being there declines from over 80% down to about 10%. And the paper also found that water transparency is important, that wild rice does much better when transparency is greater, when the water is clear. And that's probably because the plant needs light to photosynthesize as it's growing up from the sediment through the water column to get to the surface where it can have floating leaves. And also remember that wild rice needs cold winters, and the warmer the water, the less likely it is to be there. So those are the 3 variables that paper found that were controlling wild rice presence across Minnesota. And this graph shows how sulfide and transparency work together on the probability. You can do multiple binary logistic regression, and the surface shows that the highest probability of wild rice being there, 90% probability, is when the transparency is very high and the sulfide is very low. And you get really low probability when the sulfide is high and the transparency is low, less than 10%. Remember there's 64 variables measured, and some of them correlated with the presence of wild rice, but the paper argued that it was just sheer correlation. For instance, phosphorus and nitrogen go along with eutrophication. The higher the phosphorus, the more algae, and the more algae, the less water transparency. So wild rice does not like eutrophic waters just because there's not enough light. And then another paper, the 4th paper, was headed by Kurt Pohlman. He was one of the peer review panelists, and he got involved in the project. He performed the structural equation modeling, which is trying to understand what's controlling the sulfide. PCA had thought that it was controlled by the iron content of the sediment. But what Kurt showed was that it was simultaneously controlled by 3 variables. You can't make sulfide unless there's sulfates, that's one of them. And the bacteria don't work unless there's organic carbon to eat. And on the other hand, once the bacteria makes the sulfide, if there's iron available, it removes the sulfide from solution. So he showed in that paper that these three variables were equally important from a statistical point of view. And interestingly, back to Merbo paper, those three variables, no one of them is actually statistically significant on its own. Even though you can't make sulfate without sulfide, or you can't make sulfide without sulfate, the sulfate concentration isn't statistically related to sulfide. And organic carbon is also not, and the iron is not. So all three of them together in particular combinations determine how much sulfide is in the mud. And here's a graph relating the sulfate in the surface water from all the sites that the Merbo team sampled in the field to the pore water sulfide. And you can see that when sulfate is low, down to 1 milligram per liter, you really can't make much sulfide because there's no sulfate to make it. But as the sulfate increases, gets higher and higher, sometimes you make a lot of sulfide and sometimes it doesn't make a lot. And if it doesn't make a lot, it's because there's not any organic carbon for the bacteria, or there's a lot of iron, or both. Sometimes it does make a lot of sulfide up here. These red dots are when there's no wild rice. The blue dots are where there is wild rice. And these gold triangles are some samples from commercial cultivated paddies that the teams did just to see what was going on with them. And then actually the last of the 4 articles is another one headed by Amy Merbo, where her field crew sampled John Pastor's mesocosms and looked at them a lot more variables. And they found that not only was sulfide made when there's a lot of sulfate, but the bacteria were digesting the organic matter and releasing what was in the organic matter, which was the nutrients, phosphorus and nitrogen, and also the mercury that was attached to the organic matter. And because these bacteria also are the primary bacteria that methylate mercury, they made more methyl mercury. Methyl mercury is important because it's the only form that accumulates in fish. So this paper demonstrated that adding sulfate to a water body fundamentally changes the ecosystem through the releases of the phosphorus, nitrogen, mercury, and more methyl mercury. So PCA proposed a revised standard and had to define the beneficial use and the number to protect the beneficial use. And the beneficial use is the wild rice water for the harvest and use of the grain as food. And the PCA identified about 1,300 water bodies that would be protected by the rule. And more could be added later. And the PCA identified a sulfide concentration that would be protective of wild rice of 120 micrograms per liter. The Minnesota Chamber of Commerce supported a study, but it came up with a much higher number, but the PCA thought that was too high. And so back to that graph relating sulfate to sulfide, the protective concentration was identified as 120 right here, but you couldn't control it just by controlling sulfate. You had to do something, either control sulfate or organic carbon or iron, but you couldn't. The organic carbon and iron are part of the ecosystem. You can't change that. So what the PCA proposed was figuring out how much sulfate would work with the organic carbon and iron in each individual water body to keep sulfide below 120. And they proposed doing this by calculating that sulfate concentration using an equation. And this equation produced really different numbers for different systems depending on the organic carbon and the iron in that system. A lake called Little Round Lake had high carbon and low iron, and the standard would only be about 1 milligram per liter because more than that would make too much sulfide. But a site with a lot of iron could have a much higher number of 186. And then the PCA looked at how often that equation would be wrong. Sometimes calculating a number where it was either the sulfide would be much higher than that or lower. And it called that the misclassification rate. With the equation, it was 16%, but the best you could do with a fixed number was 32%. So it was twice as accurate as a fixed number. And the best fixed number, interestingly, was 10. If you had to pick a fixed number, it was 10 milligrams per liter. And that's the best number because it's wrong. It's too low 16% of the time and too high 16% of the time. And then there was complicated administrative procedures that the PCA had to go through. There were 6 public meetings, 4,500 written comments, had to respond to all the comments. And then the administrative law judges oversaw the whole thing, had 30 days to prepare a report. And there was many comments received. For instance, 10 milligrams per liter should be retained because any increases in sulfate could enhance eutrophication and the methylation of mercury, increasing fish contamination. Or that sulfate greater than 10 would produce black iron sulfide plaques that interfere with nitrogen uptake. Or that the protective sulfide concentration is not stringent enough, or other people said it was too stringent. Or they said the PCA's list of wild rice waters is too broad or the list is too narrow. And it went on from there. I'm gonna hurry up because we're running out of time, but you get the gist. There's a lot of objections. And then the administrative law judge issued her report on the standard. And the PCA had developed rules dozens of times prior to this one. This is the only one that was totally disapproved. And why did the judge disapprove it? Well, it made front page news in the Star Tribune. She said that she had to make sure that the rule was demonstrated to be needed and reasonable. And she wrote a 82-page report with 321 numbered points. She concluded that the PCA had fulfilled virtually all legal and procedural requirements and that the science-based objections had been convincingly addressed, except for the black plaque on the roots. Nevertheless, she concluded that the equation-based sulfate standard is supported by peer-reviewed science and is needed and reasonable, but she said the equation is overly vague because the results of the equation are unknown at this time. The wild rice waters list did not include all the waters that the DNR had put in their 2008 report or the waters nominated by tribal organizations. And the PCA did not evaluate impacts of the increased sulfate on other pollutants downstream like phosphorus and mercury. So that's why she disapproved it. She had other important points like the Native American community did not believe the rule would be protective and they did not feel that they'd been heard. And she noted that there were a few sections of the proposed rule that were not opposed. And she said the equation-based sulfate standard is not rationally related to the agency's objective because the data wasn't available to insert into the proposed equation to calculate the individual standards for each wild rice water body. And then she said, therefore, if the rule is enacted as proposed, there would be no standards when the rule becomes effective. Regulated parties would not know what the standards were that would apply to them. Therefore, the rule as proposed will not protect wild rice from the impact of sulfate and is not rationally related to the agency's objective. And it's unconstitutionally void for vagueness. A rule like a statute is void for vagueness if it fails to give a person of ordinary intelligence a reasonable opportunity to know what is prohibited or fails to provide sufficient standards for enforcement. The PCA appealed in March and was denied. And then the PCA withdrew its proposed revision in April. And the PCA had responses for all these 3 major objections, but the judge and the chief administrative law judge did not agree. And that's the end of the story. We have a few minutes for a couple of questions.
Okay, thanks very much, Ed. So we got a bunch of questions. I guess we'll just try and get through them. If you have to go before we're done, there won't be a presentation next week, but there will be in 2 weeks by Jessica Hellman on conservation interventions. And then I'll just go ahead with these questions. So first question, how did Moyle and others measure sulfate at the time? And how well do these older measurements mesh with modern analytical methods?
I think the method that was available at the time was a barium precipitate that had a detection limit of about 1 milligram per liter. But I think the numbers are accurate as measured.
Okay. Wild rice in 200 parts per million sulfate waters, were the sediments anaerobic?
Oh, virtually all sediments are anaerobic. Yes.
What's your explanation for why there are still healthy wild rice beds downstream of the Boswell power plant?
When the crew went and sampled, they collected the data that could be used for the equation, and they were there 3 times, and the 3 numbers that were produced were about 40 milligrams per liter. So the standard would have been around 40. By coincidence, that's what the hearing officer set.
Okay. Next one's a long question. I was a student at U of M Duluth a couple of years ago. I did a risk analysis for the potential for sulfide from wild rice lakes to flow into the St. Louis River using GIS and data you had given me. At the time, I didn't have a complete understanding of what happens to sulfide when it's transported, or even how it is transported to the river. So I was left with a lot of unanswered hypotheses. Do you have any insight to the potential risks that the copper sulfide mining may pose to the neighboring wild rice lakes and ultimately the St. Louis River?
Well, sulfide is going to oxidize the sulfate, and it's going to leak out of the system, and it's going to get into different systems which have different vulnerabilities. I think what the PCA has trouble with is having a holistic standard. It really regulates one metric at a time, and so sulfate has a number of negative consequences, and PCA was just paying attention to the wild rice at this point. But I think it's serious. The mercury methylation is serious, but that's even harder to understand than this effect on wild rice and the relationship between sulfates and mercury methylation is really complicated.
Okay, next question. Has any research been done on which bacteria convert sulfate to sulfide, or is it all bacteria? Could there be landscape differences in bacteria communities that affect the transformation of sulfate to sulfide that influence different success rates of wild rice in different areas?
Was that about mercury methylation?
That was about which bacteria convert sulfate to sulfide.
Okay.
And could there be differences in the landscape in those bacteria that might affect the rates that they do that and how well wild rice persists?
Yeah. Yeah, there's several groups that convert sulfate to sulfide, and they work differently, and the whole microbial ecosystem is very complicated and beyond me.
Okay. Is there a database available with comprehensive measurements of sediment, organic carbon in Minnesota lakes?
So far, just for the lakes that were studied for the study, and maybe a few new ones. I think the Pollution Control Agency has been collecting this data regardless of what the judge said. So that there are new data available. I don't know that it's all in one database right now, but it could be requested.
And then from the same person in a follow-up, it seems to me the equation could still be used to estimate or assess the vulnerability of different lakes to sulfate in a watershed planning context. Is the data being collected and made available by the PCA?
Well, right. I think the data is being collected, especially for wild rice waters. And any data that PCA collects is available. It's public data.
you think the administrative law judge made a mistake in her holding, or is maintaining the 10 parts per million standard reasonable?
Well, I think the PCA got really interested in the scientific accuracy of the standard. It may well be reasonable to use 10 from a practical point of view. It's just that it's not right all the time. So how do you deal with it being wrong part of the time, and how do you admit that and not have it be an unreasonable standard when you know that it's not right? But all standards are wrong some of the time. I think that the public needs to understand that these are bright lines that are drawn across complicated environment.
Okay. Do you see the PCA moving beyond single substance regulation into a more holistic standard?
Not that I've heard of, but it would be really challenging, but desirable. It's not the way regulatory agencies are built.
What portion of lakes that were studied or used to develop the equations for a sulfate standard would be at risk at the sulfate standard of 10 parts per million?
Oh, okay. A standard of 10 milligrams per liter results in about half wild rice waters being at risk, because the standard ought to be lower than 10. So about half should be lower.
Okay. And Amy Mirbo shared with everyone that the data from the papers you mentioned are available freely, including organic carbon measurements. The next question, do you foresee the PCA enforcing the 10 part per million standard in permitting going forward?
I don't know. I don't work there, and when I retired, they stopped talking to me. So I don't know. I don't know what the plan is.
With regard to polymet, the PCA has indicated that it plans to adopt a site-specific standard that would presumably allow polymet to discharge more sulfate than the current standard. Oh, sorry, that's a statement, I guess, not a question.
I think polymet agreed to meet the 10. That's my understanding.
Okay. All right, well, I guess we're right at 1 o'clock. So thanks so much, Ed, for a very informative talk. And thanks everyone for attending. And we'll see you again in a couple weeks.